那个啥，有的时候不会自己隐藏。
它那也，它也别的，
这个东西，那个有很多。这个来。
对，是。
完了。
我
用的指针。那指针放。
打针。
指针最重要。
老师，赵老师必须让用指针。啊，可以，都打不好。OK。
开始吧。
我这还没来呢。赵老师没来。
他这个，
他说他要到现场。
हाँ ये कसम से तो मानेंगे भी नहीं आप जितना मर्जी का कहेंगे आपने ठीक है यही कहता है ये भी है
नहीं ये प्लस है ये थोड़ा प्लस करना है आपने ये तो सेवन
数据改成了
有一类
他是搞工作的，不用的人看就会。
对，共享那个共享。
Yeah, so that better translate the group.
好像。啊，2点。
到了。
介绍吗？
对，介绍一下。那就知道。对。
开始。
Good afternoon, experts, faculty members, and students. I'm Zhao Hua, the presenter of today's seminar. It's my honor to share with you some intriguing research findings regarding MCD. The title of my presentation is MCD Spectral Response of Phosphorylated Amino Acid. The content of this presentation is as follows. The first section is magnetic field and the origin of life. Professor Zhao has long been dedicated to research on the origin of life, and this book is about phosphorus and the origin of life. I highly recommend everyone to read it. Phosphorus play a very important role in the origin of life and evolution of life. So, as no life would exist without it. And magnetic field conditions, how does the phosphorus would influence the chiral selectivity and the alpha and beta and gamma amino acids? Next, we will focus on addressing these questions in our research. Let me first introduce the CD and MCD. CD is a circular cyclonism. It's a spectroscopic in which chiral molecules interact differently with circularly polarized light. It therefore gives an idea about the conformation of a molecule and its change upon changing in conditions. And the MCD provides information about the spin and orbital angular momentum associated with ground and excited electron states. Here is a picture. The picture is about the Jessico 1700. There is a box. When you open the box, you can see there is a permanent magnet of 1.6 Tesla. You can change the direction of by your hand from north to south direction and south to north direction. The measurement range of this instrument is from 185 to 2500 nanometers and 190 to 400 nanometers represents the most frequently utilized region. The scanning speed is about 100 nanometers per minute, allowing the accurate data collection. We should notice that for chiral molecules, the obtained signal MCD pair is the sum of the MCD and CD signals. Thus, actual MCD equals to MCD pair minus CD signal. But for achiral molecules, the MCD pair equals to MCD signal. In our previous study, we found that phosphorylated alpha amino acid can therefore assemble into protein-like structures and react with nucleotide to synthesize nucleic acid. However, the phosphorylated beta and gamma amino acid, along with their structure analogens such as sulfur and carbonyl, lack this property. Then, with the instrument of MCD in hand, we are ready to test the MCD of the amino acids. First, we tested three simple amino acids such as alpha alanine, beta alanine, and gamma and this gamma aminobutyric acid under the standard MCD measurement conditions. Notably, none of these unmodified amino acids exhibited detective magnetic MCD signals, indicating that a lack of magnetic receptive electronic transition in their non-phosphorylated state. Next, we synthesized and tested three phosphorylated variants. The experiment revealed that only this DIPP-L-alanine have obvious MCD signals, and its beta or gamma counterparts remained inert. The secret may lie in that alpha amino acid it can form a 5-membered ring transition state. The hybridization of phosphates changes from sp3 to dsp3. I think this discovery may explain why nature might have favored alpha amino acid and as less building block. Why phosphorylated beta and gamma amino acid didn't have MCD signals? Because when we add 1 or 2 carbons to this amino acid, the beta and gamma amino acid will form the 6-member ring or 7-member ring. However, they are energetically unfavorable, but the alpha amino acid could form 5-member ring transition state is energetically favored. Our findings reveal a critical selectivity in magnetically response behavior. Only phosphorylated alpha alanine generates a detectable MCT signal, where its sulfur substitute and carbonyl modified analogy show no MCT activity under identical experiment conditions. This data contrasts tell us that the unique role of the phosphate group CD and MCD phosphorylated alanine is very different. We can see that the MCD signal is very beautiful and symmetrical. This red one on the top is positive under the direction south to north, and the black one is negative under the north to south direction. With a distinct peak emerging at 180. Wait, 这好像没电了。Wait, sorry. At 187 nanometers. But how we can explain this 187 nanometer MCD peak? In our previous studies, our computational CD analysis demonstrated that glassine derived bicyclic spiral compound has a peak near 186.6 nanometers. This signal predominantly ranging from a pi to S. Orbital transition in the nitrogen to phosphorus bond, followed by the electronic contribution from the long pair of carbonyl oxygen group, and finally the sigma capital component of carbon hydrogen. The above results were obtained from Professor Wang Yuefei at Shanxi University. The similar wavelength of DIPP-ARA and the pentacoordinate hydrogen phosphorine of diglycine, their peak is very similar. So we suggest that they may be shared a same mechanism, where the MCD signal in phosphorylated alpha alanine likely arise from this the hyperconjugation system of phosphorus reduction, carbon and carbonyl moiety, and the magnetic quantum number of phosphorus with CSP3. Hybridization is plus and -2. It's magnetic sensitive. Okay, that 为什么总是没电了？
什么？对面。
Okay. Okay, let me show you the real pentacoordinate phosphorine derivatives. 好了。
电池要换一个。
Okay, let me show you the real pentacoordinate phosphorine derivatives. Both the phosphorus and the phenylalanine have the chirality generating 4 isomers. The first stereoisomer has the lambda phosphorus configuration paired with R amino acid. There is a symmetrical MCD signal at 187.9 nanometers and 200 nanometers. We should note that this compound is a pure compound obtained from column. Then we synthesize the aryl pentacoordinated phosphorine containing alanine and phenol, followed by test its CD and its MCD. The experiment results show that it has a distinct absorption at 193 nanometers, demonstrating remarkable consistence with previously observed electronic transition patterns in pentacoordinate phosphorine system. Under magnetic field, this sp3 hybridized phosphorylated alanine could form a dsp3 hybridized five-membrane transition stage. It has a MCD signal at 187 nanometers, and the other real pentacoordinate phosphorine has similar signal around 200 nanometers. I think they are closely associated with this pi electron transition within this hyperconjugation system of phosphorus, nitrogen, carbon, and carbonyl moiety. As we know, this phosphorylated amino acid can form a high energy five-membrane transition stage capable of undergoing electrolysis and nitrogen to oxygen transfer and nitrogen to ester transfer reactions and so on. Our current research demonstrates that only phosphorylated alpha amino acid could exhibit MCD signals under magnetic field conditions, which explains why life has selected phosphorus and alpha amino acid. Next, Professor Johnson will display the critical role of nitrogen phosphorylated molecules in biological process. Finally, we thank all the people contribute to our work, and thank you for your patient listening. That's all. Thank you.
Okay. Good afternoon, everyone. I am Dr. Song Fu, and Dr. Zhao Hua has introduced the significance of phosphorus chemistry, right? And the unique features, especially MCD, that is a light magnetic combining technology to characterize the properties of compounds. So we'll keep going about phosphorus chemistry, and I'm focused on the protein phosphoryl transfer mechanism, and some just ongoing preliminary data in our lab. Okay. First, regarding to the protein transfer, we want to say, I think most of the people don't know. Actually, ATP binding protein is the oldest protein on the Earth. And about 3.7 billion years ago, the ATP binding protein has been emerged. And then after that, the phosphotransferase, okay, mainly constitute of kinases that emerged. So actually the life is normally organized based on phosphorus chemistry. And more specifically, I want to say that ATP is a very, very important. I believe all of you know this molecule, right? Adenosine triphosphate. And it's very interesting that the daily ADP conversion of a person undergoes equally their body weight. Is it amazing, right? Why? Why we must need so many ATP turnover one day? Because maybe one of the most well-known function of ATP is acting as energy currency, okay, to synthesize protein, genes, and as RNA synthetic material and a cofactor for enzyme. Maybe this is not that familiar for you, but I want to focus on the phosphorylation donor. This is the key point of this topic. And I want to say, how does ATP act as a phosphorylation donor? Actually, the kinase is the key player, the main player for phosphor transfer for a life system, okay? And more importantly, and more regarding to the chemistry, the mechanism we're talking about, that's I want to say that the key catalytic residues in the kinase catalytic pocket is a base and an acidic residue. And no matter what is the phosphoryl acceptor, imidator, phosphoryl, guanidine, carboxyl, hydroxyl, any else, the conserved, key point of the catalytic activity depends on a base and an acid. So you want to say, what is the role of these two kind of amino acid residue? So there is a popular catalytic mechanism or conventional kinase catalytic mechanism, that is acid-base catalytic mechanism. That is, these two amino acids act as a general acid base, just like in organic synthesis, but this is a water system, right? And the detailed mechanism is that when a kinase, this is a kinase residue, basic residue, acidic amino acid, and this substrate, for example, hydroxyl group as an acceptance receptor, and the gamma phosphate, how to transfer from ATP to the substrate. So people will say, okay, the aspartate, okay, it will act as a base, from here, this one, aspartate 1066. It will deprotonation of the hydroxyl group, then it become a nucleophilic fire, okay? And then it will attack the gamma phosphate of the ADP and it transfer. So people always call it a ping pong mechanism, just to put it on and accept. But the things truly like this, we're gonna have a discussion from a chemist perspective, okay? So we all know that when you learn the study organic chemistry, you will know amino acid, amino and carboxyl group actually is much more stronger nucleophilic group, nucleophiles than hydroxyl, right? And this is not that strong basis. So why lysine and aspartate in the conserved pocket, why not they be phosphorylated, right? You might ask this question, and I will told you, right? You're right. During Professor Zhao's about 30 years, investigation on this field, we have found from a small molecule model for this transfer mechanism, just like this. Actually, Dr. Zhao has previously showed this picture, right? And actually, detailing when an amino acid, the alpha amino group are phosphorylated, it will undergo a very magic 5-membrane intermediary. And this intermediary is very active and neither nucleophilic group or nucleophobic group can attack it. For example, when the hydroxyl group are aside near it, it will go to from this mixed hydride to this O phosphorylation. That is the hydroxyl group. And also, when there is a phosphorylation of carboxyl group, it can form a carboxyl phosphorylation, okay? And so on. So you can see from here, actually, from a chemistry perspective, you will see, okay, the alpha amino group is in a base, right? The alpha carboxyl group is an acid, right? So actually, only amino acid, actually, it has all the characteristics of the kinase, right? So we say the nitrogen phosphorylated amino acid is a mini kinase or a pro kinase or a kinase from the very earth, on the very earth stage, when there are no life, only ATP, it can act and function as a kinase, right? So in this model, we can say both the nitrogen phosphorylation and the carboxyl phosphorylation anticipated in this transfer process. So we might ask, maybe the kinase phosphorylation transfer process involved a more detailed mechanism, but normally people do not see it. Maybe first the nitrogen phosphorylation, then form this one, a penta-coordinate phosphorylation intermediate, just like this, and transfer to the ultimate substrate. So you might ask, do nitrogen phosphorylation and carboxyl phosphorylation really exist in life system? I will answer you, yes, exactly right. Till now, actually protein phosphorylation is one of the most important and extensively studied post-translational modifications on the life system. The other, might be acetylation, ubiquitin, and others. And people know, we already know there are 4 kinds of phosphorylation. There are oxygen phosphorylation related to the serine, threonine, and the tyrosine, and the nitrogen phosphorylation, histidine, lysine, arginine, and carboxyl or acyl phosphorylation, aspartate and glutamine, and cysteine. But why people, especially the biologists, never known the importance of nitrogen phosphorylation? Because the nitrogen phosphorylation are really, really not stable, especially acid and heat label. So the routine experimental workflows, especially in the biological chemistry, this kind of nitrogen phosphorylation are always degraded, so people never seen it. And the over progress, I will not say the detail on it. The Dr. Li Fulai will give you a more detailed data. Actually, they are found very early, even earlier than oxygen phosphorylation, but the people know little about it. That's because it was difficult to study. So the first question we faced at that time when I was a PhD candidate in Professor Zhao's lab, first question is, does nitrogen phosphorylation really existed in a life system, especially the eukaryotic cells? We want to know what is the quantity, distribution, and function of this kind of modification. But there are some challenges, especially the acid heat label property and the low abundance. So at that time, I studied to make some solutions towards this challenge. There are 3 tools: antibody, proteomics, bioinformatics, to help us to gain more deep understanding of this kind of modification. So first is an antibody-based, we develop an antibody. Just we saw that this bond, PN bond, it's very unstable. So how to cheat the life to help to produce antibody for you? So there's an elegant way. Just change the PN bond to PC bond, but the electron configuration is also similar to original PN bond, right? So when this antigen injected to animal, it will produce antibody for you. And successfully, we developed this kind of antibody, including phosphoarginine and phosphohistidine, not shown here. And we use it to see that really, does human cell really contains nitrogen phosphorylation? And the answer is yes. And we found that this kind of cell, Jurkat cell, contains a much more higher abundance of nitrogen phosphorylation. So keep on going. We developed a phosphoproteomic workflows to reveal the nitrogen phosphorylation in Jurkat cells. And I won't show the detailed technologies, but we have some solutions to solve the heat and acid label property of this kind of modification. So finally, we have identified over 100 of both three kinds of nitrogen phosphorylation: arginine, histidine, and lysine. And from bioinformatics and analysis show that we can see that both either arginine, histidine, and lysine proteins are mainly involved in RNA processes. For example, the mRNA splicing, mRNA exporting, and others. So, and a more detailed bioinformatics analysis show that these nitrogen phosphorylated proteins are disease associated, such as the breast cancer, the colorectal cancer, and virus infection, and the other disease related. So actually, as time goes by, more and more groups in the world notice the importance of such kind of novel PDMs. Okay, so, and in addition to us and many other scientists, such as Tony Hunter, the Kinase Kim, and other scientists that began to follow this research, and more sites are obtained in the world. So we began in a lab, the Dr. Zhao Minxiao in the lab began to collect these data and establish a phosphocyte database and predictors. And from at that time, maybe 5 years ago, she found that while over 60 protein phosphorylation database and predictors exist, but only a few are protein nitrogen phosphorylation specific. So she developed the first database containing all three types of nitrogen phosphorylation data all over the world. And it's a website you can click on. You can just put on your data, okay, your ID, protein ID. You click on the search. It can tell you the PDMs and other informations. And from mining the data, we found a very important message that is among the nitrogen phosphorylated proteins identified in the experiment, there are over 70% also exhibit oxygen phosphorylation site, suggesting a potential correlation between the two. It means that a crosstalk. And notably, there are over 100 proteins contains all three types nitrogen phosphorylation. So, okay, back to our initial question: what is the matter regarding to the phosphor transfer mechanism? So the question is, we know that many proteins have nitrogen phosphorylation and oxygen phosphorylation, and both are existing in some proteins. So naturally, we can think about that. What is the dynamic phosphor transfer processes between these three kinds of modification, right? And also, we think about some solutions. That is from the database, nitrogen phosphorus database, and the AI tools, AlphaFold3, to give us a broad, give us an overview, mining of the data with this such kind of PDMs. And then we choose some interesting proteins to do wet chemistry, a wet experiment, such as mass spectrum-based proteomics and NMR experiment. Okay, and also the data are so many, big data, you know, over 10,000 phosphor sites. But which site will choose? So there's also an elegant point, that is the buried phosphor site. You know, when the site, phosphor site was buried in the protein, you might think, oh, does the kinase can attach it, access it, its substrate? So there might be novel chemistry, you know, novel phosphor transfer mechanisms buried in it. So we also, from a big data, 40,000 phosphor sites, and with the solvent accessibility analysis, there are only about 1,000 phosphor sites, buried site. And then we, using a Nature paper, they have collected all of the motifs of the oxygen phosphorylation, and we exclude these kind of kinase motifs related to phosphor site. And then there are only about 100 phosphor sites. This site, we call it naturally phosphor transfer, not just by any enzyme. This chemistry must be similar to our nitrogen phosphorylated amino acid model, right? I told you before about the small molecule. So, the functional analysis show that this protein class mainly contains enzymes and other proteins. And the goal enrichment analysis shows that they are mainly related to the metabolic process, especially nucleotide. So it's very important. And we also show you a very unique analysis. That is a 3D motif, not similar to 2D motif. That is, we want to see a phosphosite in the 3-dimensional view. Which amino acid residue are nearby it, you know, not just by the sequence motif. So from this view, we can give you a conclusion. That is, the acid-base frequencies near the buried site are significantly higher than that of the enzymatic sites. What does it mean? Okay, it means that it was very similar to our small molecular model, right? So for this kind of site, naturally, they are a kinase, even though they are not a kinase in the biological classification, but they own the kinase, you know, activity, right? So I'm gonna show you two cases. We're interested in proteins. One of it is ROA. This protein catalyzes hydrolase, function as a hydrolase, and it hydrolyzes the FBP, this kind of phosphorylated sugar, to smaller molecule, DHAP and G3P. And we're gonna see how does and we have found that among the in the catalytic pocket of this protein, there are a buried phosphosite that is phosphorylate serine 271. So we wondered how does this amino acid can be phosphorylated, and very lucky they are already answered in the existing PDB bank. And this PDB bank, you can see, right? This is the substrate. That is this molecule. there the molecule 1,6 biphosphate sugar, right? So you can see it just directly the phosphate transfer from this substrate to this serine. And it's very interesting, not by a kinase, but just by its substrate. It can capture the phosphate from its substrate. So we call it intermolecular evidence or intermolecular phosphate transfer mechanism. And another, we're also lucky too. There are another PDB bank. We can see there is a phosphate group. Just there nearby there's a serine 2022 and histidine, and they are like both of these two amino acids can form a chemical bond with this phosphate. So you can see this is an intramolecular evidence that the phosphate can transfer from protein, from one of the amino acid residue of the protein to another residue nearby. So from this, we can establish the two kind of phosphate transfer process, that is intermolecular and intramolecular, right? And another case, this is the kinase, okay? Its name is NME1 or nucleoside diphosphate kinase A or NDPKA. Okay, from the NFOS database, we can see that this kinase, there's a residue 118 histidine can be phosphorylated, and many others such as phosphorylation of oxygen phosphorylation, ubiquitin, and others. You can check it on the website, okay? And so we want to say, and here the AlphaFold 3 is a useful tool for us to find the magic, to find the mystery of this kinase, okay? We simulated the kinase with a phosphorylated serine, and very interesting, nearby it there are histidine and a carboxyl group, glutamine. So, and they are very close, from a chemistry view, you see they can form a phosphate transfer from both three. They can, both maybe on a delayed strategy or direct. So one step, you can from the nitrogen phosphorylation to the histidine to the oxygen, or the carboxyl to the oxygen, or first from nitrogen to the carboxyl, and finally to the oxygen. So the most important, how to prove it, and I'm gonna show you a mass spec-based technology to see if this is possible, okay? So the protein, there are two groups, the control group, the protein incubation with the water vehicle, okay? And another incubation with a phosphor source, maybe an ADP, maybe it's substrate, 1,6 biphosphate sugar. And then by bottom up mass spectrometry technology, we can see the quantitatively identify if there are any phosphosite, the level of the phosphosite are elevated or something. And the results are exciting, okay? For this protein, RA, you can see we have identified a lot of phosphosite, and especially there are carboxyl group can be phosphorylated, okay? And more importantly, when compared to the control group, when the protein incubated with the FVP, that is its phosphorylated substrate, and the carboxyl group and the serine group, their phosphorylation level are elevated, okay? So it confirms the hypothesis of the intermolecular phosphorus transfer mechanism, okay? And more significantly for this protein, NME1, we also identified a lot of phosphosides. And especially interesting is that there are also nitrogen, oxygen, and the carboxyl group. All of them can be phosphorylated. And without ATP, their level is very low. But after the ATP incubation, they are almost, over 1,000-fold increase for their phosphorylation. And more importantly, these three kind of phosphorylate residue, they are all very near for each other. So we have the reason to say that there are potential, intermolecular phosphorylation. That is the phosphate transfer from histidine to the glutamate and to the serine. This is just like a fish swimming from one residue to another residue, just in the kinase catalytic pocket. It's so interesting, right? So in a brief summary, I'm gonna say take-home messages. Till now, we have some conclusion. That is the novel protein phosphoryl transfer mechanism on proteins. There are two kinds of transfer process. One is intermolecular, another is intramolecular. And for the intramolecular, there might be one step or two step. And the one step, two step, of course, need further confirmation. We are still ongoing, such as the NMR technology to do a time course dynamic, determination of their levels. And more important, I want to give you I think, because I introduced two proteins, and there are many other proteins. One, NME1 is kinase, but the other one, ROA, is not a kinase, right? So I'm gonna show you that the kinase is not a kinase, but non-kinase might be a kinase. ROA is not a conventional kinase from a biological view, right? But it can function as a kinase. So I want to raise the new definition for kinase, a general kinase, just from the view like our small molecule model. Just a nitrogen phosphorylated amino acid can function as a mini kinase. Why not other protein with acid and a basic amino acid with such kind of, circle environment, microenvironment in the protein? It can be a kinase. So that's interesting. Why? Because from an application perspective, oxygen phosphorylation kinases are important drug targets. Till now, there are 518 kinases in human cells, okay? And over 100 drugs in the market target to kinase. They respond to over a $100 billion market. So it's very important for human health, So obviously, a deeper understanding on catalytic mechanism of the non-kinase or a new kinase scopes, including other non-conventional kinase, will definitely open new avenue on drug development, right? So this is what I wanted to show you about some research in our lab, okay? So thank you for your attention. If you have questions, please. So you have any question, you can talk in this time or after the meeting, okay? Yeah.
Hello, everyone. It's a great honor for me to be here to make a presentation followed by Dr. Fu. I will introduce a study work about large-scale phosphoproteomics for protein nitrogen phosphorylation identification. Protein phosphorylation is a reversible and dynamic post-translational modification. And for a long time, phosphorylation on serine, threonine, and tyrosine was thought to be the only relevant forms of phosphorylation in organisms. Besides, over the past years, besides the commonly recorded oxygen phosphorylation, the phosphorylation can also be occurred on other non-canonical amino acids such as aspartic acid, glutamic acid, cysteine, and lysine, arginine, and histidine. Due to the phosphorylation occurs on the side chain at the nitrogen atom, so it's also referred as nitrogen phosphorylation. Nitrogen phosphorylation found earlier, about 60 years ago. However, their instability renders its study. Unlike phosphoester bonds involved in oxygen phosphorylation, the phosphoramidase bond involved in nitrogen phosphorylation, the high TB free energy of hydrolysis of phosphoramidase makes them more labile and sensitive to acid and heat. And conventional methods and protocols for phosphoproteomic study often involve several harsh conditions, especially for digestion distorting and phosphopeptide enrichment. So in the past years, great efforts have been made to use the for nitrogen phosphorylation identification, especially methods or tools used for nitrogen phosphopeptide enrichment, such as antibody tools. However, now existing antibody tools are limited to use for phosphorarginine and phosphorhistidine protein identification. Besides, several novel strategies such as molecular imprinting strategies or fast-test trap strategies were also developed. However, these methods only can be used to one type of nitrogen phosphorylation identification. Besides, implied metal affinity chromatography and the metal oxide affinity chromatography are the two most used methods used for phosphopeptide enrichment and phosphosite identification. By optimizing the solution pH and shortening the incubation time for phosphopeptide enrichment, these two methods have been successfully used for nitrogen phosphorylation identification. In our previous study, Dr. Fu have been used the titanium dioxide methods used for phosphopeptide enrichment at mild acid conditions and used for nitrogen phosphorylation site identification. As a result, nearly about 500 nitrogen phosphorylation sites identified from JAK cells. Besides, other studies based on those methods also used to nitrogen phosphorylation site identification. However, the nitrogen phosphorylation site identified is limited, so the prevalence and the function rules of nitrogen phosphorylation in mammals remains largely unexplored. And this knowledge gap is mainly due to limited research tools available and absence of appropriate large-scale workflow for nitrogen phosphorylation identification. So we developed a large-scale phosphoproteomics for protein 9 nitrogen phosphorylation identification. Given the poorly understanding of the natural nitrogen phosphorylation phosphatase and lacks of effective inhibitors. So in the cell lysis process, the denaturalized buffer was still used. Besides some key process, we also optimize, such as we increasing the amount of sample preparation to enhance the scale of analysis. And second, avoiding the temperature above room temperature during the sample preparation. And at the desorting process, we use the low temperature and the mild acid conditions. And the fourth, we're using high pH to fractionating the samples. The fifth, we use a two-round enrichment, two-round phosphopeptide enrichment. Briefly speaking, after the first round enrichment, a second round was performed using a supernet of the after the first round enrichment. Before performing large-scale phosphoproteomics, we first verified the nitrogen phosphorylation in cells on microscopic level. The results show that a substantial number of proteins undergoes nitrogen phosphorylation in cells. Followed, we optimized the methods used for nitrogen phosphopeptide enrichment. We compared the effectiveness of three methods that used for phosphopeptide enrichment in mild acid conditions. So as the results show, the titanium dioxide methods exhibits a high specificity for phosphopeptide enrichment in mild acid conditions. So in the following study, we're using titanium dioxide methods used for phosphopeptide enrichment. We combined two-round enrichment strategy and high pH fraction. We developed a large-scale phosphoproteomics used for nitrogen phosphorylation identification. As a result show, the first round and second round enriched phosphopeptides, the number was compatible and showed largely overlap. Most importantly, the two-round enrichment significantly enhanced the identification of phospholipid sites. About 30%, the new site was identified in the second round enrichment. Totally, we identified over 8,000 nitrogen phosphorylation. Though the nitrogen phosphorylation is lower than oxygen phosphorylation, however, the total number of nitrogen phosphorylation sites is equally to the sum of phosphorylation at serine and tyrosine. So we can conclude that nitrogen phosphorylation is also a relevant form of phosphorylation in human cells. We further analyzed the credible of those identified nitrogen phosphorylation sites. About over 40% of those sites with high confidence. So as a result, over 3,000 high confidence nitrogen phosphorylation sites were identified in MC7 cells and active cells respectively. We further analyzed these proteins, the subcellular localization of these proteins harboring high confidence sites. Result shows those proteins are predominantly enriched in nucleus, followed by cytosol. And protein molecular function enrichment analysis shows these proteins are enriched in chromatin bonding, histone bonding, protein domain spec bonding, cadherin bonding, transcriptional co-regulator activity, and ATP-dependent activity. And the cellular component analysis shows these proteins serve as the components of nuclear envelope, centrosome, and the nuclear spike. And geobiological process analysis shows those proteins are predominantly enriched in chromatin organization and RNA or DNA metabolic process. We further analyze the amino acid sequence of those nitrogen phosphorylated peptides. The results show that nitrogen phosphorylation peptides exhibit a conserved phosphorylation motif. The serine and the proline will frequently occurred and nearby the nitrogen phosphorylation site. And some several significant motif characters associated with nitrogen phosphorylation were detected. Previously, we observed that the serine is enriched nearby the nitrogen phosphorylation site, so we further analyze the phosphorylation state of these serines. We found that over 60% of serines will also undergo phosphorylation. So we guess there may exist some crosstalk between oxygen phosphorylation and the nitrogen phosphorylation. In our study, we further verified that the histone phosphorylation of ME2 protein at a site of 180 facilitates the transport of phosphor group within ME protein and between ME proteins. Further, we applied this workflow used for tissue samples analysis, and we performed proteomics and phosphoproteomics. As a result, over 8,000 proteins with quantitative information were identified in 3 pairs of colorectal cancer tissues. About over 300 proteins were up-regular in tumors, and these proteins are predominantly enriched in EMT process, which is associated with tumor metastasis. We also observed that some kinase and phosphatase about that regulated histone phosphorylation. The expression level of some kinase and phosphatase was the expression level in tumors was changed. So the kinase and phosphatase, the expression level changes may cause the phosphorylation modification levels change. So phosphoproteomics we also performed on these 3 pairs of tissue samples. Totally over 2,000 of nitrogen phosphorylation sites were identified in 3 pair samples. Based on label-free quantitative analysis, about 448 phosphor sites with the modification levels change more than 2 times were identified. This means nitrogen phosphorylation may be involved in tumors. The hallmark and KGT enrichment analysis shows that these proteins are enriched in adherent junction, tight junction, and apical junction. Particularly, about 43 phosphor sites with modification level change over 2 times were identified in all 3 samples. Particularly, MAP1B, TJP1, and CTNL1 and TLN1. These proteins functions are associated with cell junctions, and this protein expression levels expression levels in tumors will also change, and which may disrupting the cell junctions. Loss of cell junctions, tight junction barrier functions, and also associated with tumor metastasis. So this nitrogen phosphorylation on these proteins may also involve in may also involve in the cancer metastasis. So that's all. That's my presentation.
The group regarding original life. And yesterday, Dr. Huang on the hierarchy. But today we talk about nitrogen phosphorus. So maybe I could summarize a little bit for you so you could understand all the connections, what we are doing. And we are not only we are looking for the original life, but also for the today's life, also for space life, right? And we would like, we are also, although we are a small institute, but we have diversity faculties. Faculty like Professor Liu Xinming, space life medicines. Also AI expert, Professor Chen Yuzhong. And we have analytical experts, Professor Huang. And so we could integrate all this. So maybe the topic is looks like very diverse, but however, we have some more specificity on the phosphorus chemistry. So I could quickly give you some overview. Maybe you already read from the all the speakers from the day before yesterday. 我坐着讲也行啊，他们面上可以照到吗？可以的，我共享。共享啊。Okay. So I think for the original life on the Earth is very old, right? 46 God before, and where is the life again? Maybe at the very early beginning of when we had the Earth formed. They think about maybe already had origin of life formed. So they call it the hidden area. In the hidden area, just very beginning of the formation of Earth, or they say maybe we have the Dukkha. Dukkha is our ancestor, the life for the ancestor, right? And the many, many important oxidation events also could make our life get more and more complicated, right? So I think last time in the Dr. In group already talked one of the original life theory is called about the thermal, Vander thermal, or maybe they call it the iron nickel cluster, sulfur cluster. And this is a cluster. I think many of the organic experts, they know that they use the clusters to do very fancy one carbon chemistry. So our original, we could think about is one carbon chemistry through one carbon. So on the Mars, they be only carbon dioxide there. And it could Mars have died or not? The CO2. So from either CO2 or monocarbonia, they could form a very active chemistry forming on the cluster. So the cluster could form a fancy organic chemistry, like forming acetic acid, or forming this 3 carbon source. If there's amine there, the amine interact with the 3 carbon chemistry, forming alanine. So we have many people talk about alanine, right? One of very important amino acids. So they could form in the peptides. In other words, this iron nickel cluster could form in peptide library. So how is the nucleoside formed? They say in the space, HCN, 5 HCN conforming the adenine. HCN is very toxic, right? However, 5 of them forming adenine. But the other theory is that if we go to the how our biological system in our body forming AMP from where? From AMP from the ribose, just ribose, the sugar, the diphosphate, like one of the examples Dr. Fu talk about the 1,6 the diphosphate, the ribose, they could form it. They could add in one of the glutamine to give the nitrogen for them. And then second amino acid glycine, glutamine, and then the other aspartic acid forming the AMP. So from here, it means in your body, we didn't eat the nucleic acid to form in our body's nucleic acid. We eating we are not eating proteins. The protein digest, give amino acid, then amino acid forming the nucleotides, okay? So in other words, it means maybe the formation of the nucleotide didn't from the outer space, right? It is from the amino acid, the metabolism system. So this chemistry means very supportive. Life might start from amino acid, right? And then they could form in a very important system. And ATP, Dr. Fu talked about, the oldest protein is ATP binding protein, and the evolution of this fancy protein needs the metal ion. In our body, all the animal needs all the life, we need the metal ion. And the metal ion relate to what? Like Dr. Zhao Hua said, we have the magnetic field. Even although we have very low magnetic field on Earth, only 53 microtesla, very small. But on Earth, even thousands less, the magnetic field. So magnetic field is very important. So like Dr. Lee, he used if you sharing the magnetic field and capture the cell there, the cell behave differently. They are sensitive to the magnetic field. So in other words, magnetic indeed very strongly. And this may be due to our enzyme. Our enzyme need all the metal ions. And LUCA, this some of the people say that LUCA, if the LUCA should start one carbon chemistry, like acid. The bacteria may be the oldest, one of the oldest bacteria on the Earth. Because if you have acetic acid, right? And the LUCA need a lot of systems. And the minimal system here, amino acid metabolism, and then you have the nucleotide metabolism, and the ribosome to form peptides. In other words, the ATP importance is different. You already know that, right? The LUCA is a bioinformatics says, oh, maybe just 0.2 GA or 0.2 GA. It is very important. That time we have the LUCA. Some people so much earlier than what somebody expected, right? Now we go to the metal ion. The metal ion is so important, okay? Metal ion, and on the early Earth, very, very early time, we have a lot of ion so far. Also coincident with the ion cluster, right? So far ion cluster. And indeed, at that time is the reduced form. In other words, we don't have much oxygen at the beginning of the Earth. It's a reduced form. So reduced form, the ion is +2 charge, not +3. So there's a, Dr. Fu also did a lot of experience, compare +2 and +3, or these two ion difference might do ATP synthesis or ATP hydrolysis. Yeah, they have make a difference. And iron 2 sometimes more effectively. So that means, or iron 2 has much stronger magnetic susceptibility. So they have some relationship with that. For example, this one of the example, and Dr. Hu's group doing that, right? Use the acetyl phosphate. In other words, without the ATP, how the ATP could form at the beginning of the Earth, maybe acetic acid, this mix and hydride. Acetic acid could form from the hydrothermal, right? Like we mentioned, one carbon before, then convert to acetic acid. And the acetic acid react the phosphate, phosphoric acid, the mix and hydride. And this small reagent could convert ADP to ATP. And among all the metal ions, the real iron 2 and is more efficient than iron 3. That Dr. Hu's group's doing that. So in other words, at the very beginning, where's ATP come from? It's not from our cells, no cell at that time, okay? It could from the inorganic, catalyzed by the inorganic ions. And this metal ion, some are more strong magnetic susceptibility, the other now weaker, or either paramagnetic or anti, right? So this metal ions play a lot of role for our origin of life. It's connected to the magnetic, we call it the intrinsic, okay? And this inorganic, like inorganic phosphoamine, this is Dr. Huang Bili group doing that. Just under the treatment with urea, urea also could form very easily, very beginning under thermal conditions, and they could form ATP. So ATP not necessary from the cell, not from the life. Inorganic form the ATP, and it depends on the condition. And phosphorus is so important, right? This is the law, the Tao the state. If life exists anywhere in the universe, you should consider whether they have the phosphorus. And so we are focused on the phosphorus role. And in other words, when the Earth at the beginning, we have all different materials. Why choose amino acid and the alpha amino acid? Dr. Zhao Hua gave you a little bit of idea, right? The difference of alpha, beta, gamma amino acid. And only alpha amino acid was chosen. And the chirality only chose the L amino acid. So these are the chirality, the difference. So we are doing all this under the phosphorus chemistry. So mostly we are set up this very interesting model. This is one of the ribose there, and one of the amino acids there, 5-5 spiral. And this is the top secret model. And we already set up it many, many years, okay? And the story here, if we had beta amino acid react with butanol, nothing happened, nothing. The phosphorus NMR give always one peak. However, if you have the butanol, they have forming exchanged quickly on the phosphorus. The phosphorus signal changed. In other words, just the structure difference. And the difference, Dr. Zhao Hua told you, just one carbon away, it's make a big difference in the chemistry and the structure. And they are so, one is sensitive to the magnetic field, the other no. So this is not it is the structure determine our life, okay? And the calculation could also prove that the alpha give you 5 memory, more energetic fabric. 6 memory is very, very impossible to form, okay? And the next question, why nature chose the ribose? We have amino acid story talk about, right? But how about the ribose? The ribose is very interesting, right? You need the 2 + 3 + cystiol. And this cystiol is so important. And Ashimoto is the very famous, carbohydrate chemist in the world. And he thought about that maybe hydrogen bonding is very important. So he set up many very fancy nucleosides. And however, he found some of they could form very strong hydrogen bonding, but are not chosen by nature. Our nature chose this one, not chose the one, not this structure. So we need this structure. Why? So we have our own reason why. So we just mix them. This is chemistry, right? And the amino acid could quickly form the spiral. And the story, we're going to chirality, okay? The chirality, Zhao Hua show you all the structure, right? It's interesting. If you have amino acid D and L at 2 isomer, right? The phosphorus chirality. And here we have the pentacoid and the phosphorus chirality is theta or lambda. 2 times 2, you have 4 isomers. So these 4 isomers, I would like to show you that even though you have the amino acid, S maybe L amino acid, right? You have S carbon, but the phosphorus, if you have delta, they have the positive carbon effect. If you have the lambda, it's negative carbon effect. In other words, doesn't matter where your amino acid, left or right, no, L or D. The phosphorus chirality, he is the one controlling the chirality direction, either positive carbon or negative. So in other words, this is the importance we would happy to announce in this meeting, that the phosphorus chirality controlling the lives chirality, the structure, okay? The positive carbon, okay, this is one of the most important slides I show you here, okay? And then why nature chose the L amino acid? This is a Nobel Prize winner, Dr. Ada Yunus. And she said, why our ribosome? This is a very big, fancy system. And they only recognize the amino acid, one chirality, the L amino acid. And Dr. Ying Jianxi here, he is the one so smart. Just adding 3, this inorganic phosphorus compound, okay? Inorganic phosphorus compound, adding with the nucleoside, and the amino acid use the 1:1 ratio, 1:1 L and the D. But that's nitrogen label. Nitrogen label the lactogen, the L amino acid, 1:1, and then this is dirty in where? In aqueous, in water. Okay, quickly mix 3, then they connect together. They connect to this fancy molecule. The molecule with 1 ribose and one nucleoside and a phosphorus and the amino acid. If you select the nitrogen 15, this phosphorus split to doublet. So you could do in 1-part reaction to identify the chirality selection. So the second question, how come on Earth we chose the L amino acid? And indeed, if you use the D ribose, then they chose only L. However, if you reverse, that is reversed. If you change the nucleoside chirality to L, that is not chosen on Earth, somewhere in the universe. Then they choose D amino acid, okay? So this is the result from his experiment here. We didn't go too much detail, okay? Anyway, this is the Chinese Kung Fu, right? Chinese Kung Fu means all these are done by the Chinese student and the Chinese faculty. Yeah. So we call it Chinese Kung Fu. And in other words, if you D amino acid, they love the other word, L nucleoside. If you have L amino acid, they'll choose D. And this is happen on Earth. So it should be on Earth, we have magnetic field. And our Earth, we have a direction, rotate, right? And our Earth following rotate around the sun, our galaxy also rotate on the galaxy. They have only one way, not 2 way. So they chose only one. If like a magnetic field, right? If you have rotate, you have direction, you have vectors, and they chose only one vector, and that's why. If you choose, if you let another universe, or everything reversed, the rotation around the Earth, Sun, or then the galaxy, then they might choose different. So we think there will be a second. They say where it will be the second Earth. That they use similar, maybe use a similar compound, but the chiroid is converted reversed. Okay. And then we talk about the reason why. So we would like to show you that 2-dimension NMR is very important. We could have the first dimension NMR at phosphorus with the proton, and then if we label the amino acid nitrogen 15, the phosphorus could split. Okay. And you could see, indeed, this is a true structure because the phosphorus could couple with all the protons on the nucleoside. Proton 2, 3 plus couple with that alpha amino acid, right? But also, this part is the amino acid couple and the nucleoside. So the phosphorus connect one hand with the nucleoside, one hand with the amino acid. And here could give you the story about the chiroid dissection. And this is go to more complicated stereochemistry. In other words, if we have the 2 chiral, right, 5-5, one is ribose, one is nucleoside amino acids. You could put the day on the side. I show you here. The amino acid, if you on the equatorial, you have 2 ways to put them. If on the axial, you have 2 ways, or the other. And among all the 6 possible isomers, only the first 2 are most stable, and the other are not so stable. So we have to do all this. I won't show too much detail, but anyway, for this, like Chen Su, she did the serine. A serine has the genetic code among 16 genetic code. So another the AGCU, all they are depends. Serine depends. So he has shown that still he showed the intermediate activity. And then we have calculation. In other words, like the chiroid on the phosphorus is very important. And also amino acid. Here, if you have a D-uridine and the L amino acid, the chiroid on phosphorus is the delta, but the energy is much less than the D amino acid chose. So we could do the experiment together with the calculation to put the chiroid dissection. Second, also the genetic code origin. It's prefer which one has lower energy, and we experiment will show that. Okay, this is non-published, but I would make the conclusion that this, the story here, this is model, like the Zhao Hua tell you, that this SP3, but it could be go transition state DSP3, and we have all these models going on. But finally, go to the model with the nucleoside, and that's the top secret to link the 2 together to study the genetic code origin, and then also study the chiroid selection between these 2. Okay, so this is very important that we have our small molecule and here. And the kind of story Dr. Fu Songsen told you that this is they show you before here, and this lady, she was in the spaceship there. She's not come back, still there. They have we have the laboratory near there. She's going there. So this NPOS is very useful for, we would suggest for you, just these 4 characters. You could go to this database and then you put the name of the protein. They will tell you all the story about the protein, about their phosphorylation site, nitrogen or oxygen or carboxyl. They also tell you the story about oxygen phosphorylation, or tell you about the methylation, also ethylation, ubiquitination, everything. So this database gives a very large information, save you a lot of time, okay? And so next is the metal ion is so important. Magnesium, naturally the nuclear number is 24, 25, 26. And the day 25 is paramagnetic. And the 24, and they are inert. However, if you give clearly separate all these three isotope of magnesium and their effect on the, for example, the ATP synthesis or ATP synthesis, the magnesium 25 indeed could help a lot. In other words, the magnetic field, this is caused by the metal ions. Metal ions also help you. So the metal ion, just at the beginning, I told you that this is very important for all the life. The life needs the metal ions, okay? And the magnetic number, if you have d orbital or f orbital or any like, they could have resonance more, then they have the magnetic number even larger than 0. Or if you have 0, then there's no magnetic quantum number, then they are inert to the magnetic field. However, if you have the higher the magnetic number, then they are sensitive to that. So like Zhao Hua told you that, okay? And that's the story I don't want to repeat here, right? She showed you this story here. So our story today is that we study the phosphorus chemistry, starting from the origin of life. We could very beginning, hierarchy selection, genetic origin, then to today, the protein chemistry. The protein phosphorylation, non-phosphorylation, and this is indeed a connect, very important, the center of the life. So we would like to give you all this introduction so you can know what we are doing here, okay? Okay, thank you. And any question, you could ask any question here, okay? To Dr. Yin Jianxi, also here. Anyone? Any question? So we could discuss today, and the tomorrow discussion would talk about what you are interested in to discussing more in the future or cooperate more in the future. Tomorrow morning, 9:30, Professor Liu also come. Dr. Yin, also the other. But this is the time for issue of this day. Any questions? You have questions? No? Do you, I think, do you understand what they are talking here?
Yes.
Because we are from the biochemistry, so all those reactions we are already teaching to our students.
现在有没有那个三maybe yesterday is because to rush, and then they have very fancy 3D chromatography, and we have saved some time. You could introduce me to it. 听那个，哎，黄丽玲介绍的。Because we have also very strong magnetic NMR. This morning you are there. This 800. This 500, 400. So NMR is also our rely a lot. So Dr. Huang, would you please give them a little bit more detail of the 3D? That's very important.
但是我得跟你说，
没有关系啊，不是新的技术，可是它用除三位，
这是能出
那没有关系啊，那你们做嘛，你讲吧。
昨天后来给他们补充了呀，
补充。
后来我们那个 BB 讲完有单独跟他们。
是吗？那你再介绍。So you have any question you interested to ask Dr. Huang. 你介绍一下，或者你有别的东西，你仔细讲一点吧。啊，儿时你有什么说法？
就是您今天跟黄老师说那个OIC，
黄老师对啊，你们那个是
他去市里开会，
是他做的。
他去市里开会
对
去了。
对，你慢慢讲吧。他们昨天问的问题，你们解答了吗？
解答了。
解答了，那很好啊。
这个其实不是核磁技术，只是人为我们用那个软件给他做成
哎，
这样的。
对，那你跟他们说怎么做的嘛？
跟他们说过
说过了。OK。So you have anything maybe other than lecture?
But
Yeah, you have any questions?
in lecture,
Yeah, you have questions? Yeah, we could more informal. Yeah. Any question?
when he described the reaction of aldolase, one of the speakers, when he described the reaction of aldolase, that is the hydrolysis, lysis reaction of fructose 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. That is the lysis reaction. Though the mechanism involves the transfer of phosphate at the catalytic site of enzymes, but there is no transfer of phosphate because both phosphates are already present on the substrate. But mostly kinases involve the transfer of phosphate from ATP to other proteins.
Yeah. So he just activates them more.
So he's interested to keep that aldolase into the class of kinase. Kinase is a transferase class,
Yeah,
and the aldolase is in the lyase class.
Yeah. The kinase actually changed from ATP gamma to the other substrate.
Mostly kinases involve the transfer of phosphate from ATP to other protein, like in the signaling pathway, you have a serine threonine kinase, tyrosine kinase. So these kinases involve the transfer of phosphate from ATP to certain proteins for their activation. But in case of aldolase, the fructose 1,6-bisphosphate already have two phosphate, one at the position 1 and other one at the 6. And when it's split into a keto and aldose sugars, 3 carbon sugars, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So there is no addition of phosphate. Just the catalytic sites involve the transfer of phosphate for the reaction, but still there is no transfer of phosphate on the substrate or product. So is it right to put it into the class of kinase?
Okay. I mean, you are right.
你可以
totally
放
right.
放回去
Yeah.
给他看。
Yeah, you are so professional. Yeah, that's a very good question. You are right. There are two phosphate, right, group, and after the catalytic process, there are 1 to 1, small molecule, and another one. So I mean, actually, the catalytic process maybe not for this protein, but for the PGM kinase. PGM is also a kind of protein like this. It's also catalytic. That is the isomerase, 1.1 phosphate sugar change from 1 to the 6 point.
Yes.
And the phosphate, the phosphate was captured from the PGM to also a serine or a histidine. And then there's a flip 180 degree, and then the phosphate from the transfer from the the PGM, this enzyme, also an enzyme, to the substrate. So it's the isomerase transfer from 1 to 6. But for this,
One
Yeah.
phosphate to glucose 6 phosphate.
yes, you're right. And for this protein, actually, it's not that clear for this, the phosphorylation of this histidine. But I mean, this the phosphate, we are not really know the biorelevance, the biosignificance of this kind. But we found it can transfer from the substrate to its own, to the enzyme.
And then transfer back to the substrate.
Yeah, but in addition to that, the nearby serine histidine can also get this phosphate. That's why. And this is a new chemistry, but we don't know what is the bio, function of this kind of, process. But maybe in some times, maybe it's a, auto feedback, when the substrate, the concentration of substrate is, very high. So it will inhibit the kinase, the activity of this protein, of the enzyme. Maybe just like this, it's a very old theory, many, the pathway, circle pathway, there must be a feedback.
So in
When-
future, there should be a modification of the definition of classes of enzymes.
Yeah, you're right. So it's interesting, but right now, in the current stage, we are focused on the chemistry. But the next step, we are trying to answer another question, why? What is the biofunction? So yeah, thank you. You're a very good question. Yeah.
Question I need that.
I just had one question, it's not related to this. I just wanted to know the people who are working in the field of studying protein ligand interaction. I asked this question of people when we are visiting the NMR lab, but those are working on studying protein carbohydrate interaction.
This morning.
Yeah. And when you're the lab here, they are also studying using this for the structural analysis of small molecules. So I just wanted to know, is there any people who are using the NMR for studying ligand protein interaction, like STD NMR, model of the transfer?
NMR study the protein structure in the actin molecules.
Yeah.
And Dr. Billy, she used that before. And actually you should label the protein as the 93.
Yeah.
Now we know all these techniques.
Yeah.
All of Dr. Henry's morning, he's expert in the carbochemistry. No, to the protein structure for the memory.
Yeah.
For the memory structure.
Yeah.
But that's in general. In general, he labeled the C13 of the membrane. But it's all right, we have this instrument available.
Yeah.
We could do the protein in that the ligand, also metal ions. We are
Right.
interested.
So much, but
We have the Dr. Wang before doing that.
okay.
Yeah. PhD.
I was doing that. And also, someone molecules produce proteins using eukaryotic system or prokaryotic system.
The protein they could.
Production.
I think that they could, all the system they 你们可以找吧。从
Sorry.
Section of
the protein
recombinant
capture from
recombinant protein production use the eukaryotic system. So there
As an expression system.
Yeah.
who is working in this?
So you may, who is working on it?
Yeah.
Who is working on recombinant protein?
production.
not the native proteins. The
The
native proteins are existing in nature, and she's asking about recombinant proteins by using vectors, by the
In the
insertion.
cells, like this, because I produce them in prokaryotic system, but I are doing it in eukaryotic system, like human embryonic kidney cells, like this. Is there anyone around who works in this field?
Actually, it has mentioned NME1, this kinase. It has
Okay.
yeah, chance action.
Guys produce here.
Just similar like that. It could go like.
Like you mentioned, NME1,
Yeah,
NME1 use the vector produced in the human cell.
yeah.
And then we use that study the phosphorylation
Maybe I
site.
can have that contact details of my
这里不对，他说人的细胞能够培养。Yeah, we could, any of the protein we want, we could. Like then use the NMR to study the cell. Yeah, that's I think it's a routine technique. They can do that. And then we are interested to know whether you other than oxygen, nitrogen, or carboxyl, or sulfur. Okay.
Okay. I would like to have this or part of
Okay. That depends which protein we specific. Pairs. Then to know whether this NMR. Actually, we have already cooperated with the most basic NMR in China, in Beijing, by Institute of Chemistry in Beijing. And they have many regulars NMR. However, they can do the functions. In 800, we could do up 600. The progress. And they are very strong idea.
Yeah, we also have the 800 biomarkers in our institute and
Yeah, we have the others. And we have debated with the most other one, most higher power. And then they are going to next week.
Yeah.
The passport site. If you can separate it on the 800, other than 600, not far distance. And they use the tracking and you can check which nitrogen, which position, or oxygen, or carboxyl. They are very like, big area difference. Yeah. We are happy to cooperate with them. In China, we have very strong structural biologists.
Yeah.
And we are more interested in the phosphorus related.
Maybe we can collaborate in that because I asked for but then the other people. The professor, I think, and other people, they all are together.
So if you write the protein's name, they will not get but then we know this protein. It comes like it has shape. And we would like to check on the box to see whether the nitrogen phosphorylation. If the protein name, we can check it right now. Protein name, 你怎么用哪个本子去？你没有笔。You write the name and then 你们现在给它上 enforce， 现在 demo， and then we will see whether your protein has nitrogen phosphorylation.
Okay.
If yes, we are interested to know the function of your protein. Yeah, and that will be one of the important cooperation. Yeah. The enforce that the name is.
Monoamine oxidase B, a protein that is involved in neurological.
你现在 Yeah, and now this database we have actually every year. That's also version before. So the story is that the function of protein could be migrate from one position to another. And then nowadays, However, the story could go almost, forward, say, before possible, maybe 90%. And then you could say this by this, we have out here, you need a carboxyl group. You could mutate that,
它就是
then
蛋白酶化。
start this,
蛋白酶
this migration. And also know the function. 它有这个名字吧？
有。
它叫什么？如果很普通就有了。对啊。
哎呀
So any protein you want to check?
We can check it.
Check the
I have, yeah, we can.
If you have other protein interested, also write down. They can check. Give them yeah. And then interesting.
You are only studying 20 standard amino acids. Now the number of standard amino
一点
acids,
收录到，现在没有。
selenocysteine and
会 check it.
lysine.
Okay.
Yeah,
These are
same stash now it's
amino
empty.
acids that are now
Okay,
Maybe it's not.
standard amino
okay,
acids.
it's
They
not-
are boosted.
There I'm working because I am not focusing on this aspect, phosphorylation aspect,
Right.
but I want to express this protein
Okay.
in the mammalian cells, like human,
Are you
embryonic,
new amino acids?
embryonic.
Are you studying this
Like
new
you
amino
remember, I think you know this HEK system, right?
Okay.
The hex system. And I have seen the expression of this protein is reported in yeast.
These are the 21 and
So
22. in
are you
human.
guys
There are 21
working
standard amino
on
acids and
yeast
in that
or the mammalian
there
cell
are 22 standard
expression
amino acids.
system?
In
We
human,
didn't
selenocysteine
do
is the
that.
new
You don't do this.
We can do 这个 we can do hack.
Selenocysteine
You have this.
and
Yeah.
Okay.
But
and
use
cysteine.
the
So it's a modification of cysteine, and in that it's a pyrrolysis.
large amount of
That's the new
Yeah.
standard
maybe
amino
it
acid. Though
It is very costly,
the-
not only costly, it's also challenging. So I have another protein that you can check is USP7, that is
Selenocysteine
ubiquitin,
is an amino acid.
ubiquitin,
I know that.
Pacific,
Okay.
Selenium.
protease,
We call
SESENO.
7.
No, yeah.
I'm also working on this, but I have not checked it with the with is it
Yeah, we are
phosphorylation
interested on that.
purpose or not?
It is very
So,
important. for
I'm
the
looking on so many
right?
protein, but I
Stop mechanism,
I
right?
just wanted to know if you guys are working
So you-
on producing them in system other than the prokaryotic
This is
system.
the standard amino.
So you wanna
Yeah, yeah.
so you
It's possible. You guys
There are
can
20
be your
amino acids.
email ID or your
That's the new-
profile. I will check that, and then I
I'm
can
working on proteins
discuss
like
with you.
transferrin,
You can give me your email
folic
ID.
acid receptors and
You both can give me, then I will
We are in-
write that what exactly I want to know.
okay.
Study the evolution of.
So you wanna you do structure
你
biology.
叫那个
Yeah.
So
But
what is the
小
difficult
小胃跟
why prokaryotic? Because then PDMs, I know that
We are in.
eukaryote expression system can restore the PDM phosphorylation and many others. But prokaryote we are lost
We have
such
Yes,
What
yes, PTM.
modification.
Okay, I am
Yeah.
writing using
seven
those
enzymes,
targets
they are
for
the
which
five
there is
peroxide,
no
the main
Yeah.
antioxidant.
I have selected.
So
those
this
that-
amino
You
acid is
can-
also the
okay.
part of this enzyme.
The HMA.
So it's a new amino acid. So I
So I'm
I
not
was
right now
asking that
working on
you, are you studying the and phosphorylation of this amino
We
acid?
can use the
in your lab.
mammalian system so that we can also produce those
Yeah.
proteins that have TM in
yeah.
that.
Yeah.
So right
Do
now
you
I'm
wanna-
limited to work on those proteins that do not need any PTM.
I see. You want a protein with activity,
Yes.
not loss of activity.
Yes, because some protein I have, so far I have worked on
Yeah.
Because of
5
the
or
strong
6 proteins
attack,
which do not need
then the
PTM
sulfur.
for.
So
Okay.
So now I also wanted to learn that's how I can produce protein using that, because this protein when I produce it in prokaryotic system it was
So
Mm.
those
producing,
proteins are
but
reductases.
its functional activity was not there.
Reduced.
It was not functional.
But we would.
So this is the just I wanted to know if you guys are also working
Anyway, we could.
on this field. I can have an idea. So you guys produce the protein by yourself, or is there is someone else who
It's a
produce it for
strong
you?
nucleophile. As when we are
Use by
synthesizing
Ourselves. So maybe I
So
can
there is more
hereby I can
compared to the
see what's your paper and then I can discuss
and
further.
you use lin, it's
Okay.
a
I
more
can
stronger
see your
than
papers and
sulfur.
then
But we are
discuss.
not in structural biology. We
You are in
have to use
structural
some
biology.
protein to do some, to do some unique investigation,
We have
such as phosphorylation and such like that.
Okay,
So it's
and I'm working the ligand protein. So maybe we can collaborate in this way that you find out this aspect
have a
and
large
I
group
can
and
help
diverse
find
area
out
of
molecules that can enhance or decrease the
the
function of that
biochemistry
protein.
are also
Maybe we can
doing
see
kind of
it in future.
Okay.
So, but here you just
yeah,
showed
because of
us the
antioxidant.
phosphate NMR. Yeah.
Yeah.
So you are mostly doing that because you are interested in phosphorus chemistry.
Okay.
Okay. So this is good, but can you kindly write your names as well?
Mostly free radicals.
Your names, your name and your name.
Here.
So these kind of
This is
can be used as a
this is
preventive.
name.
This is a family name.
Okay, Lee and then Fulai. Okay.
Family name Fu.
So you are
Okay, thank you. I am Sumaira.
we always have the set.
Your diet is very good. You are mostly using.
I'm
ghee
from
and green cheese, mostly not that the green
ICCCPS,
China is the
Prachi University.
common in
Yeah,
China.
it's MIT.
You might know that she has collaboration with Professor Dr. Ishmael.
We are
So I'm his student.
Yeah,
Right now,
Your
working as
lifestyle,
professor.
but still your lifestyle and diet habits are better than us.
With?
But I
You are.
You are student.
hot water.
I'm
Esophageal
student
cancer is
Professor Dr.
very common in China now.
You know, she knows that.
Okay.
And I'm
No, I
right.
think
I mean, okay, you send us.
Then
We can
see the
check
check.
You can.
你发现了吗？上去了吗？
Thank you.
算出来，但它这个没有收录。
没有收录这个办法。
我们肯定
We check it on this database.
Yeah. You want checking your.
We will check it on it.
Yeah,
These are.
good database. We will use it.
I think you are also a member of the group who generated this database.
So
At least by a form of PhD.
That's
So I saw that you have written 4 university name book. Yeah, Chengdu University, Chengdu University also. Yeah, yeah, Xiamen University.
even your student resembles to you.
Yeah. Okay. Then any more questions?
Thank you. Thank you.
Okay, thank you. Then this afternoon we have meeting here. The tomorrow morning, tomorrow we here.
Discussion session.
今天你妈妈，今天今天中午9点半吧。Nice to meet you all. Nice. The schedule is 9:30. 9:30. Okay, tomorrow 9:30 here. Okay, see you tomorrow.
Okay, okay.
You know, you could decide in your mind what which you want.
Okay.
Yeah. Is more free. It's like free. And the afternoon is closing.
Closing ceremony.
Closing ceremony. Okay. Okay. And then lunch, we will have a big lunch meeting.
Yeah.
Okay.
Lunch.
Okay, see you tomorrow.
Tomorrow.
Good luck.
कौन कहने हैं ब्लैंक कहने के दिया
हाँ तो उधर
आज
是可以关个电。
为什么在没有？
但好像我们这边不是那个，他们那边。
这里
and and the meeting and the Zoom meeting.
meeting，那个 Zoom meeting 的名字出来了。Zoom meeting 呢？
我知道他们用那个
都没解怎么会的话？
他们